day everyone. Welcome to the lecture 5 of the course Biological Data Analysis and Visualization with R. In the last few lectures, we have talked about basic functions of R and we have talked about basic data structures. So, in the last class we talked about vectors, lists and metrics. These are different data structures that are available in R and we use them widely. And we also have looked into the hands-on, right, some of the functions, how do you apply to these different data structures. One data structure that we have not discussed so far is the data frame, and this is one of the most important data structures for us, because you will see most of the biological data comes in this data frame format. And in the subsequent classes, as we will do hands-on with different biological data, you will see these most of them are of data frame type. So, either some of them will be might be in matrix type, but then again most of them will be in data frame type. So, in this class we will be talking about this data structure. This the last data structure that we want to talk about is the data frame, and then again we will talk about some of the functions that you can apply on this data frame. Some of the functions are common with what we have seen for matrix data structure. So, here again we will see some of these functions for data frame, and they are pretty similar, but there are also some extra functions that we will talk about that we can apply to data frames. And then followed by that we will talk about file input output. Okay, this is very important in case of our analysis, because most of our data will come from some experimental output, right. So, we do some experimental assay and we kind of get some data out, and then we want to process it, right. So, this is where this file input output part will be very important. Okay. So, how do you do that? We will see this in this class, and we will take again the hands-on examples, right, and I will illustrate how we actually do this. Okay, so these are the topics that we will be talking about in this class. First, as I said, we will talk about the final data structure that we wanted to talk about is the data frame, and then we will talk about how to read and write data from different files, right. So, there are two parts again, right. You want to read files, data files that are generated by some sort of experimental assay. So, that part is the data reading part, and then sometimes we also get some processed data, right. After we have processed our experimental data in R, we get some output, right, and we want to write them or store that output and for future reference, right, maybe for plotting or for some other analysis, etc. So, we also need to do some writing, right. We generate some data and we want to write it in R and store it as a file, right, sometime a file that is possible to read us later. Sometimes we will also store the visualizations that we make, the plots that we make in R, because we want to display, right, the analysis that we have done and the inferences, right, the outcome of those analysis, and in the visual representation, and we want to store those as plots, those plots as files, right, and we will talk about that in a separate class, right, when we actually go into basic data visualization part, how we can actually store plots as files, right. There are ways to do this, and we can also control resolution, we can control the size of that file, how big that file would be in terms of dimensions, etc., the plots, etc. All those things we can control, and this is something we will be talking about when you talk about visualization. In this class we will be talking about only writing data files, right, the data output that we get after analysis in R. Okay, so let us begin with the data frame part, right. So, these data frames are tabular data that can contain different types of elements, right. So, we have these vectors and lists. So, similarly we have data frames, they are comparable to matrix, and data frames can store different types of elements, right. So, we can imagine this is something that is probably very useful in biological analysis, because we sometimes have factors, yes, no, etc. Sometimes we have, let us say, some specific types or strings that we attach, right. So, we classify samples according to some specific characteristics. So, those could be only string, but then we also get some output, right, which could be numbers, right, some signals or something, right? Those could be numbers, right? So this will be evident when you actually work with real data. So this is how we will define a data frame, right? This is what we use, a DF, and then this is, we define the data frame DF, we assign, right? That's why we have the assignment operator, you can see here, right? And then we say data frame, then we define name is this, right? See, we have some strings here in this example, some names of some fruits, and then we can have number, which we define again with this as a list or as a vector, right? So we then put them together in a data frame. Okay, we'll apply this and we will see how this works and how do you actually define data frame. Now, some of the functions that we can apply to data frame we have seen already, right? And these are common across matrix as well as data frame, right? So for matrix, we can also apply this function dimension, right? This again, it will tell us how many rows and how many columns are there. We can also find out individually how many columns are there using this ncol function and nrow functions, right? So these functions we have seen already in for matrix, and similarly we can also define row names. We can get the row names and column names out. This is something that we have also done in case of matrix. So these are common across two different data types or data structures. So then we can also access elements. We have already done in case of data in case of matrix, very similar function df. If you want the first row, then we say df1 comma, and if you want specific element, we just need to give the row number followed by the column number, right? So in this case df12. We can access columns by saying the column name as well, df $name. Okay, this is something that's sometimes is very useful, because we do not always remember which column contains which variable, right? So if we name the columns by the variables, then we can access the columns directly by the name of the variables. So again, this is a very useful command. We will see when we apply when we work with biological data. So this is something that's possible here. The other functions that we have not talked about is we can, for example, say view this data frame, right? It will show you the data frame, but sometimes these data frames are huge, right? Because we work with often with big data sets, right? For example, if you are working with gene expression data set or some sort of data set, genomic data sets, these are really huge. So you don't want to see the full data set, but what you want to see is part of the data set, right? So in that case, we use these commands head and tail. So what happens, head command will give you the first few lines, first 6 lines to be precise, but you can also specify how many lines you want or how many rows you want actually. And then you also have the tail command, which will tell you which will show you the last 6 rows of the data frame, right? So this is something again you can control how many rows you want. Okay, so this is possible, and then there are 2 other functions which we have not talked about in before in case of matrix, etc. The one function is called rbind. So it's possible. So imagine this example here where we have this data frame, right? We have 2 columns and 3 rows. It's possible to add another row, okay, and using this command rbind. Okay, so what this rbind command will do, so if you define this again these 2 elements and you want to add them here below this data frame, you use this command rbind the original data frame along with these elements. Okay, so we will again try this out and we will see how this works. We also have a very similar command called cbind, which actually can do the binding of columns, right? So like rbind, so again we have this original data frame where we have 2 columns and 3 rows, and with a cbind we can have these 3 elements and we can simply combine them right at the end of this column. Okay, so we'll try this out again in the hands-on and we'll see how this works. Okay, so this is about the data frame, very basic functions and some of the useful commands that we can use for data frame. What about then reading and writing data files? Okay, so let's move on to that, and here are the different types of data files that we can handle quite easily, right, with basic functions, but of course we can also handle other types of data which require sometimes specific packages. Okay, so these are the most common type of data files that we handle in R, but if you want to handle specific type of data sets or data formats, that can also be done, but for that we have specific packages. At least we will talk about one such package later on, and we will actually work with a slightly different data type or data format. Okay, so the file formats that we generally use is the table format usually, or CSV format or TSV format. So, you must have seen this CSV format or TSV format, and CSV format is comma separated values, right. For example, these values will be stored, right, in the data frame like this, separated by comma, right. So, in each row you will have values, multiple values that will be separated by commas in between, right. So, this is something that we can do, that we can read in R, and similarly you will have tab separated values. This is the standard practice, right. We have these values that are tab separated. Okay. So, this is something that will face regularly, right. We have to read this type of files, and in that case the command is something like this. If we are reading the table files, you can have, this is the command that we will use is read dot table. This is the command. We have to give the file name, okay, and then we have to say whether if you have a header or not. Okay. So, what this header means is that whether the first row is the names of those columns. Okay. So, this is something we will specify whether the first row contains names of the columns, or actually from first row you just have values. So, that the situation would be different, right. How do you access these variables, etc., how do you access the columns will be different. And then you can also have a separator, or this is optional, right. Usually this is tab separated, but you can also specify the separator. Maybe there is a special separator that you are using, then you also want to mention this separator. For CSV file, there is a specific command, it says read dot CSV. So, which you can take to actually, which I can use to read the CSV files, right. So, these are the common commands that we have. For writing files, we can write something like this, right. So, we can say write dot table data file. So, again this is possible, right, whether you want to use specific separators, etc., in say when you say write dot table. So, write dot table is when you want to write this data in a file, right. So, usually it is a text file or TSV file. So, you will say the data and give the file name, and then you can optional, that is an option that you can also mention the separator, that kind of separator that will be used to actually separate the data in the file. Similarly, we have a write dot CSV command, again the data that you want to write in the file. So, that data will go there, and then the file name. Okay. So, that is something we can give, like any file name that we want to give. Okay. So, there is a specific type of data file in R that is available, and that can store multiple data, right, multiple objects. Okay. So, it is not just one data that can be stored in a file, and these are called R data files. Okay. So, these are unique R files for storing different objects in R. Okay. So, if you might see this, and this is a way to actually share data very easily. You can share objects, you can save all the objects that you have generated, all the data that you have generated in one single R object, right, one single file, which will be, let us say, program.r data, and you can simply share that R data file to anyone, and then they can load it and they can work with that data on access specific data sets within that data. Okay. So, this is R data. How do you load it? So, how do you actually read it is by saying load program.r data or any R data. So, this will end with .r data file, and for saving you can save, as I said, multiple R objects, right. It is not necessarily just stored only one, right. So, you can actually save multiple objects like data 1, data 2, data 3 in program.r data. Okay. So, how do you then, if you load the program.r data, how do you find out, right. So, this is something we have to do is using this command ls that we talked about, right. So, it will immediately show you, right, what are the files that have been loaded, using this load command, right. So, you can access those, find those objects, and then you can individually access those objects and work with them. Okay, so let us now go and check out some of these functions in R, right. So, first we will talk about the data frames that you have seen, and then we will try out this the other reading and writing files. So, let's see, right. Let's first start with data frames part, and then we'll look into the reading and writing part for our data. Okay. So, here is the our console again, right. So, first we want to work with this data frame, right. So, we'll define this data frame title df data frame. Let's give some space. So, I'll simply write okay, name and let's say number. Okay. So, let's look at the data frame. So, you can simply say df, or you can also say view command. Okay. So, this is not on. So, we can use this df command for now, right. So, df command gives you this name versus number, right, that we just defined, and so the name is xyz and the number is 1410. Okay. So, we can again apply some of these functions that we talked about, is the dimension and in column, etc. Right. So, here we have, for example, in call df 2 columns. We can also try the dimension function that we tried earlier, that we have seen earlier, right. So, 3 rows and 2 columns. We can also see what are the row names of df 1, 2, 3. So, we can put row names if we like, right. We can assign row names, right. Okay. So, for example, we can say, I do not know, replicate 1, replicate 2, replicate 3, okay, and we can say now df. Okay. So, we have assigned row names. One thing that's also we can do is actually we can take row names or column names from another data frame, and we can assign to row names of df. That is also possible. Okay. This is something that also works. For that, we have to define another data frame. For example, let's say d1, which is okay. Just as an example, we are defining this. Sorry, yeah, I have missed a C command here. Okay. So, this is the command, and we have d1. So, we have now defined this. So, what are the column names of d1? Is value 1, value 2, value 3, right. And what we can do, we can define the row names of df from the column names of t1. Okay. So, this has been done, and you see now these row names actually are derived from these column names of t1. Okay. So, this is possible, and sometimes this actually is useful when you are manipulating data, biological data. Maybe you want the gene names here and there, right. Those kind of things you can easily do like this. Okay. The other functions that we want to look at as the head tail functions, right. So, that is something we have to talk about, head and tail. Let us see. So, head df. So, it will give you, so we have only 3 elements anyway, so it does not make much sense. We will try it out on a real data, and we will see how this works. The R bind and C bind command. Okay. So, let us try this. Okay. So, maybe we will add a new row. Okay. So, this is the new row, and what I will do, I will try this. Okay. So, we have now defined this R bind, and you see this has actually now been added, right. There is a new row now added at the end of DF. If you want to store this as a new data frame, you have to again use the assignment. You have to assign to a new data frame DF2, which we are defining here and assigning, and DF2 will contain this. Similarly, we can again go back to DF, and let us say we want to assign a new column, right. So, let us define a new column by this. So, new column is, let us say, okay. We just take new numbers and then C bind. So, column bind new column. Okay. So, this new column has been added. Okay. You can again change the column name, etc., however you want to define it, etc. Only thing you need to remember and need to be careful is the dimensions, right. So, the dimensions should match. So, when you are adding using C bind, you are adding a new column, the number of elements in that column should be equal to the number of rows in the existing data frame, right. So, that is why you need to define 3 values, right. You should have 3 elements in this new column, because we have 3 rows in the original data frame. So, this is something that you would have to be careful about, otherwise you will get an error. So, it will tell you we cannot apply this C bind because the row numbers are not matching, etc., right. You get an error in R. So, this is something that you have to be very careful about. Okay. So, we have seen this C bind, R bind commands, the basic commands. Now, what we will do is, now we will try reading a file, right, and see how this works. Okay. The write table again work will in the same way, and we will actually use this write table commands later in the course. You will see when you process real biological data, we will use this write table command to store the output. Okay. So, let us try the read command at least and see how to do this and how we actually get a taste of a real data frame. Okay. So, let us first look at where we are. We are here. So, I will now move on to the directory, right, using this command that we defined in the first few classes, right, the basic commands in R. So, that directory that we check the directory by get wd command. We want to move to a new directory, we use the set wd command, right. So, the set wd command, I have a directory on the desktop which says data analysis in R, right. This is where I have stored this data. Okay. And here I will now read the data frame, and I will just call it as data, and this is a tab separated file. So, it is a table format. So, the read table, and the file name is called filtered merge data is. Okay. So, for now the name does not really matter. Once we go into the data, and we will use this data set for later classes for some statistical analysis. It is a data set that we have compiled from multiple data sources, and we will use this for other hands on classes as well. And we have header in that file, we know that, but otherwise I can show you if you do not use this header command what will happen. Okay. So, let us first try this with the without header command. It will load, and let us try this head data, right. We do not want to see the, it is a big data set. So, we do not want to load the whole data, we just want to see the first few data sets. Okay. Again, so what you see is that this is a really huge data, and it starts here, right. So, first you have the gene names, and then you have some parameter values. I will explain some of them later on when we actually go into hands on using this data, what these features or factors mean and the names mean. Okay. But we have not used the header. So, which means the program R itself will add a header V1, V2, V3, V4. Okay. and then you can use these elements to access use these V1, V2, V3, V4 notations to access different columns. But since our data has the header, as you can see, right, we have the gene name and then different feature names, right, and then the corresponding values below. So, we can use the header true, right. So, this is something we need to do. Okay. So, this is something you need to keep in mind when you are using your own data, whether you have header, right, whether the first row contains the names of the factors or some specific descriptions, right, about the columns. Okay. We have used this now. So, we have shared data. So, again it is showing us only the first few rows, okay. And now it has not assigned headers. So, because we have told it, right, header equals to true, we have told her that okay, we have a header line. So, use that, right, use the first row as the header or the column names. Okay. When you define column names, one of the things you need to be careful about is should not have special characters, space, etc. Then you will get an error, especially with space. Okay. If you have space here in the header names, what will happen is that number of elements, right, that you have. So, because R actually separates out elements based on space, if you have space within one name of one feature, the number of column names will be more than the number of values that you have in these rows. Okay. So, that is something that R will report as an error. Okay. So, this is something that we need to careful about, right. You should not have use space, you should use, for example, the underscore or maybe dot or etc. The other thing you will notice when you load data in R is that the header, if the header contains special characters, R will convert all of them to dot. Okay. And maybe sometimes in some rare cases it might give error as well. Okay. So, try to avoid special characters in the header line as well as space. Okay. So, this is very, very important, otherwise you might end up with errors in there. Okay. So, we have loaded this data frame. Now, we can check this data frame. For example, we have, so we can use the dimension functions, right. So, we can say, so we can see there are 2160 rows and 288 columns. Okay. So, this is quite a big data, but of course, you can have bigger data than this. You can also say, for example, what are the row names. So, row names are sorry. Okay. Okay. So, row names are actually 1, 2, 3, 4, right, numbers, but if you want to have gene names as row names, right. So, what we can do is we can do something here. We can say data 2 is using the commands that we have just described, like for all types of data structures. We can apply this data 2 equals to data -1. So, we are leaving out the first column. Okay. So, first column is the gene names. Okay. Okay. So, we can check what are the column names of data 2. Okay. So, these are the different features that are there in the data, but the gene name is now removed, it is not there. And what we can do is we can say in data 2, row names of data 2, because we want to assign the gene names as, right, as the row names. So, use data 1 as the row names. Okay. So, the first column of the data file, use that as row names for data 2. Okay. Okay. So, maybe it is giving us a bit of an error because that the first element is there in the gene. So, that is something that is probably causing an error. Anyway, let us see. Okay. So, the dimensions, so if you use this command, what you will getting is that row names length are not matching. Okay. So, this is where I think the problem is, because we also have the gene in there. Okay. So, what you can do is, let us say, okay. So, what you can do is gene is equal to data 1 So, we are assigning this in this file, and because the column name is still there, right. So, that is probably is causing the okay. Okay, so we have this data. Okay, so how many elements are there? So let's check the element number. Okay, so okay, so this is actually something to do with the length matching. So maybe we will not go there right now, but it is possible, right? So we can actually have these row names here from data, and we can see this, right? These are the numbers that we have. Okay. So we have seen this. Okay, so let's now move on to one part, right, which is just look into the data 2, head data 2, and maybe we want to write this data 2 file, right, write dot table and data 2 into a new file, right. So let's say new data file. Okay. Okay, so we can use this write dot table command. So data to new data file. Okay, and we can confirm whether there is this new data file. We can check whether that's there. We can now load this, right, as a new data and check whether there is this file there or not. Okay. Again, we have to use the header, right, because we have the header names, and we can check whether new data is there. Okay, so this is how we can read data and we can also write data. Okay. All right, so we have covered all of the topics that we have discussed in the hands-on. So just to summarize, we have talked about data frames in R and some of the functions that you can apply to data frames. We have also applied some of the functions to data frames. We have talked about reading and writing data files, and also talked about write data files, and we have seen some of the hands-on that you can do with these different functions. Thank you.